That's the way every day goes.
The first practical lab is basic concept statistical and the variation. For the objective is to calculate and describe the following descriptive statistic: mean, variance and standard deviation. Second, to understand the concept of correlation, and lastly is to determine the relationship between the two variables.
Okay, I would like to continue with our post-lab question, which is number 1 and number 2. So, first question is, discuss the differences or similarity of the mean of length between different set of leaf. Do the same for the mean of width. Okay, to answer this question, so the answer is, first, descriptive statistics were to calculate and correlation were studied between length and weight. The mean length of the leaf were different due to error during collection. The upper part of the plant had smaller leaf due to increased water loss, while the bottom part had larger one. And leaf size also influenced by age, genetic factor and environment factor. So second question is, what is mean by variance? The leaf have different measurement of width and length. What is the mean of this statement? Discuss with respect to variance. What is meant by standard deviation? The answer is, first, measure how far a set of number is spread out from their average value. The square root of variance is used to calculate the standard deviation. 3. Measure the variation of dispersion of a set of value and how measurements are spread out from the average. And standard deviation can be divided into low and high standard deviation.
Question 3. Why is it necessary to have different sets of measurement for the measurement of leaf width and length? The answer is, having different sets of measurement for the width and length of the leaf is useful in data because it allows for comparison between different sets of leaves and to ensure a consistent result. It is necessary to make assumptions about the distribution of the leaves and the relationship between width and length. By having different sets, we can compare our result with the other set. This is done to ensure that the experiment produces an accurate and consistent result. This experiment highlighted the key concept of statistic and variation. Descriptive statistics allow us to summarize and present data effectively. Measures of central tendency, such as the mean, median, and mode, provide insights into the typical or average values within the dataset. Measures of dispersion, such as the range, variance, and standard deviation, help us understand the extent of variation present in the data. In conclusion, this experiment aimed to introduce and explore basic concept of statistics and variation. Understanding variation and statistics enhance our ability to interpret and utilize the data effectively.
Practical 2, factor that affect the distribution and abundance of organisms. The objective of this practical is to introduce the simple concept of quadrat, to enhance the power of observation, to record the difference in plant distribution within the quadrant and to infer from natural observation the possible factors that affect distribution and abundance of organisms.
The first post-lab question is define the term quadrant in practice. Quadrant is some random locations where in practice should plot the area that has been chosen with quadrant that has been set, which is by 5 times 5 meter at specific area. Within the quadrant should have all trees in several types of organism to observe, the species name and the total number of organisms that have been found in the area. The method depends on linear measurements between random points and adjacent individuals, and also between adjacent pairs of individuals. A quadrant is used in practice to mark out a specific area of the plant communities to be sampled. Next question is, what are the two basic requirements of all the quadrant techniques? First one is the area of the quadrant counted be known so that density can be calculated directly. And the second one is the organism must remain motionless during the counting period to ensure that they are counted and do not go missing.
So for the question 3, the question is, what are the two basic requirements of all the quadrant technique? What would be the advantage and disadvantage of using a larger or smaller quadrant to survey the sedentary organism? So the advantage is it is inexpensive because a few materials are required, and it is a feasible and simple method to use when compared to other sampling method. So for the disadvantage, number one is larger quadrant can provide more accurate estimate of overall densities, but they are more difficult to implement due to the time required to place and sample them. And number two is, despite the ease with which the quadrant can be designed, study error may occur. So for the question 4, the question is, explain the issue concerned by ecologists in conducting sampling method. So the answer is, consider the sampling method that can be used to collect necessary data, method for selecting sample location to limit any potential bias and obtain the best estimate, and method for reducing the number of samples taken while still providing an accurate estimate of parameters.
Variations in biotic and abiotic conditions between sites may account for the variation in the number of plant species in quadrants. Among the potential causes are, the first one is light availability. Different plant species thrive in areas with more sunshine than in areas with less. Second one is soil conditions. Plant growth is impacted by variations in moisture, pH and nutrient content. The third one is competition. By out-competing others for resources, the dominant species may lessen variety. The fourth one is human disturbance. The distribution of plants may be changed by roads, pollution or logging. Lastly, microclimate. Certain species may be favored over others by variation in temperature, humidity and wind exposure. In summary, this variation result in an unequal distribution of plant species throughout the research region and are impacted by physical disturbance, species interaction and environmental gradients. Lastly, the conclusion. In conclusion, the objective of the experiment is to record the different in plant distribution using the simple concept of quadrante while observing the factors affecting the distribution of the plant and enhancing the tree observation power. The data obtained was then used to predict the true population of the area. That's all from us. Thank you.
Now baby don't hang up so I can tell you what you need to know. Baby I'm begging you just please don't go so I can tell you what you need to know. I'm at a baseball trying to go home. All of my change I spent on you.
Obligatory is an organism that cannot survive in the absence of the other partner. For example, fungi and algae that combine to form lichen are obligate symbiotic. Yucca moth and yucca plant are obligate non-symbiotic.
For facultative, organisms can lead an independent existence. Interacting species derive benefit without being fully dependent. Mutualism is not essential for the survival of either species. Both of the species will engage when both are present. For example, many plants produce fruits that are eaten by birds. Birds would be the pollinator in return.
Mutualism have two, there is symbiotic and non-symbiotic.
Symbiotic is defined as two organisms that live in close physical association. Both derive benefits, and at least one member of the pair cannot lead an independent life. The example of obligate symbiotic is mycorrhizae. Mycorrhizae is association between a fungus and plant's root. The plant supplies energy, which is photosynthesis, to fungi, and fungal hyphae take up mineral nutrients from the soil and transport them. Another example is coral reefs, anthozoans, with photosynthetic zooxanthellae. It is a trophic mutualism between coral reefs, which is coralline anthozoans, and photosynthetic dinoflagellates, which is the zooxanthellae. The zooxanthellae provide the photosynthetic products to the heterotrophic anthozoans. The coral anthozoans remove, retain, and recycle essential nutrients from the water used by zooxanthellae. The chemical interaction between the coral cells and the zooxanthellae facilitates crystallization of calcium carbonate. The zooxanthellae helps the corals grow.
Next is non-symbiotic. Non-symbiotic is the species do not live together. Both members benefit each other, and the relationship is either facultative or opportunistic. Example of obligate non-symbiotic is yucca moss and plant. This is an example of dispersive mutualism. Yucca flowers are a certain shape, so only that tiny moths can pollinate them. The moths lay their eggs in the yucca ovary, and the larva caterpillars live in the developing ovary and eat yucca seeds. Another example is acacia and plant. This is an example of defensive mutualism. Ants protect the plant from herbivores. At the least disturbance, they swarm from shelters and produce disgusting odors and attack the intruder. Acacia gives ants more than thorns to live in. She feeds them sweet sap through special organs called nectaries.
The functional response, relation between prey and predators. There are three types of functional response based on Holling 1959. There are three types, type 1, type 2 and type 3. So for the type 1 linear graph, the number of prey eaten or attacked predator increase linearly to a maximum, but then suddenly reach a constant value when the predator is satiated. Based on the Lotka-Volterra equation, dn1 over dt equal to r1n1 k1 minus n1 over k1. Best demonstrated in the lab and exhibited by the long-eared owl, Asiotus, and the Microtus wolf population.
Okay, thank you. And I will continue with second type, which is Kaitot. So Kaitot is functional response in which the attack rate increase at a decreasing rate of prey density until it becomes constant at satiation. And then Cytot behavioral response are typical of predator that specialize on one or a few prey. Example, amphidot crustaceans corophium being eaten by a shorebird, Triga totanus. Second example is the first insta of spider Linyphia triangularis feeding on Drosophila. Thank
Type
you.
3 sigmoid. Number of prey taken is low at first. Then the attack rate accelerates and finally decelerates towards satiation approaching asymptote. Predator may require a learning period to develop searching and handling skills for a particular food item before they can feed on it efficiently. Example, bay-breasted warbler, Dendroica castanea, feeding on spruce budworm, Coristonera fumiferana.